Okay, so my name is Mauchandra and I'm a PhD scholar at IIT Kanpur. Today I'll be discussing few topics of week 2. Okay, but before that, uh, I would go to explain one topic which was, a doubt for many of the students. Okay, in the previous week, there was one question in the assignment on imprinting disorders. तो मैंने सोचा कि उसको थोड़ा सा और डीप में थोड़ा समझते हैं कि what are imprinting disorders and what are the factors that actually causes imprinting. So if you recall, so imprinting means if this is a chromosome that is coming from your father and this is a chromosome that is coming from your mother, okay, and this is the position of an allele. So paternal allele, this is a maternal allele. Now, one of the alleles get imprinted or get marked for expression. So this balloon-shaped structure is one gene, okay, that is coming from the father, and it is, imprinted. But this gene, when it is present in the maternal copy, it is silenced. So when the progeny, the offspring from mating of mother and father, the child, this is a son, okay, he when he receives this paternal copy, then only the expression will happen. But the gene which is present in the maternal copy, the expression will not happen. So this is called this is what happens with imprinted genes. Okay, so certain genes are imprinted with their parental origin. But suppose what happens if this gene, the paternal copy of the gene, gets mutated somehow or it gets deleted. Then when the offspring receives both maternal and paternal copy, although the maternal copy have the intact gene, but paternal copy is missing, and that gene was imprinted for expression when it will come from the father. So so as a result, it will be like the offspring didn't at all receive the gene. The gene was not at all present in the son. So because of which the son will uh, you know express a diseased phenotype. So various common imprinting disorders that are there. One is Prader-Willi syndrome. Then there is Angelman syndrome. Okay, so these are few imprinted disorders. One thing to notice, the imprinting is happening because of the involvement of an epigenetic mechanism. Okay, epigenetic mechanism is involved. What is epigenetics? So there's a field of genetics where people study how the genes or the DNA sequences get affected by our lifestyle, like our diet or whether we are doing exercise or not, etc. So these things do affect our genes. So it can hamper the DNA repair mechanism, or it can then proofreading mechanism can be hampered, because of which few mutations can get embedded in the DNA sequence. And these mutations later on, when it is getting getting divided, this DNA sequence when gets divided, the change in the sequence can escape. So we get some change, right? That is the mechanism how mutations happen. So some of these mutations become permanent. Okay, so now these mutations doesn't have to be lethal. It can happen in a silence way. So when an environmental cause is involved, and and we are getting some mutation. So these kind of studies people do in epigenetics. Okay, so so epigenetic mechanism is there, because of which the DNA sequence of the mother or father. So in this example, the father's allele is imprinted how? Because of addition of DNA methyl groups on the DNA sequence. So now when the cell division is happening and the progeny has received both the copies, the expression mechanism, like the promoter, they will know that they have to target, they have to do the transcription of this sequence only because it is marked by addition of methyl group. So this is what happens in imprinting disorders. All right. If anyone have any query on this, do ask. I want so we have a healthy discussion on these topics, and you can be free in unmuting yourself and asking any query. Is there any query? No? Okay. Now let's move. So this was based on one of the assignment problems of week 1. Now moving to so this was the assignment problem which was given to you, right, in week 1. Which of the following statement is not true for imprinting disorders? So I told that epigenetic mechanism is involved. So because of this mechanism, allele, parental or maternal allele, the DNA sequence of that allele gets additional methyl group. Okay, the bases to the bases, methyl group is added, so it gets imprinted. So this is actually true. Okay, then so it can happen that for this example, the Prader-Willi or Angelman syndrome, the imprinting happens for a gene, okay, that codes for some protein, but it can happen also for non-coding sequences which are repetitive in nature. So this is also true. And as you see that the maternal copy was silenced, so there was some sort of loss of mutation is happening, right? The maternal copy was not being expressed. So what is not true is the sex thing, that this inheritance is not uh, specific to a particular sex. It is not like the mother will uh, will pass on these imprinted genes to the daughters. It is not like that. So for this question, option D is correct. All right. If anyone still has a doubt in understanding this particular question, do let me know. All right. Moving on. So now, for a few minutes, I'll discuss about pedigrees. Okay. So in Mendelian genetics, like whatever laws he proposed, he used garden pea plants, right? So it was easy for him to cross between male and female flowers, and as well as it was easy for him to determine the genotype of the plants by doing test cross, right? But we can't do it when we are studying genetic disorders in humans. But what we can do is understand the pattern of how the phenotype is getting passed on. So pedigree charts are made by interviewing an individual. So there's a person who comes to you and ask you certain questions, right? That my family has this phenotype. I want to know whether my children will have that or not. So you ask that person certain question. So we ask like who all in your family got the disease, means who were they married to, okay, etc. Like we ask this sort of questions. And using the knowledge that he gave us, we draw a pedigree like this. So we draw a pedigree chart, right? And then using the pedigree chart, we can give a prediction of whether or not the next generation or future generation will have the disease, right? We can give some probability of whether or not they will have the disease. All right. So this is what is a pedigree. So in pedigree, we use certain symbols, all right, to denote who are males and females, right, and etc. So square is used for male, female we denote by circle. So you are all aware of these symbols, I hope, right? If the sex is not known, we use a diamond shape. We do not fill if the person is not showing the phenotype, that is unaffected, right? Then we fill it if the person is showing the phenotype that is affected. Or if the person is dead, we cross it like this, right? And if that person is a carrier of that particular gene, right? So we give a dot. So this is how we denote all the individuals. Okay. Now, in order to show how one individual is related to another, we use this kind of like we draw lines to show that these two people are married or they have mated. If these two persons are relatives, right, we draw like two lines, means they were related somehow, like maybe they are cousins. So this is called consanguineous mating. Then if someone is a brother and sister or sister, we draw a line like this, right? Then if two brothers are twins, we instead of drawing a line like this, we draw a triangle, right? If they are identical twins, means from the same zygote, right? So then we draw a line like this to say that they are twins and also they are identical. Now, if in a family, a mother has suffered from miscarriage or abortion and she didn't knew what was the sex of the baby, and that baby has died, we show it like this, right? So this is how the relationship between individuals are shown. And we as I said in the beginning, that we interview a certain person, right, who comes to you and asks for like who about the disease, the inheritance of the disease. So that person is called the proband, who is giving you the information, who is being interviewed, and the proband is shown with the arrow like this. Okay. But this is quite easy and might be clear. This is the standard symbols that is used for uh, rep- uh, used in a pedigree. I hope it is clear till now. Okay. If anyone has any query, they can just unmute themselves and ask. Okay. Um, okay. So uh, uh, for representing each phenotype, whether it is a disease or it's a phenotype that is not affecting the survival, we have to use or we have to draw a separate pedigree chart. Same pedigree chart won't work. Okay, so you can't show the feature of rolling tongue as well as the feature of skin color, okay, who is a skin disorder maybe in the same pedigree chart. These two phenotypes has to be shown in two separate pedigree charts. Now using a pedigree chart, we can infer whether a phenotype is autosomal dominant, means the gene responsible for the phenotype, whether it lies in the autosomes, okay, or whether that gene is a dominant one or recessive one. So autosomal dominant we can say, or it is autosomal recessive, or whether that gene is related to the sex chromosomes, X, yeah, Y chromosomes, right? And also whether that gene is again expressing dominantly or recessively. Also, we can guess whether that gene is present in the mitochondrial genome, which you know is different from the uh, genetic material that is present in the nucleus. Mitochondria have its own DNA. Okay, but there are few complications. Okay, because of which complications in the uh, this pedigree analysis, uh, because of which sometimes this it is not very easy to say whether it is autosomal dominant, autosomal recessive, or X-linked, Y-linked, etc. Okay. Hmm. Now, so you are given a pedigree chart, okay, and you are asked to analyze it. So, what question should you ask? First, you should see whether the phenotype is expressed every generation, okay, or whether it is skipping any of the generations. Okay, first you have to look for that. Second, you have to see uh, whether that phenotype is getting expressed in both sexes, both male and female. But your analysis is not just stopping by answering these questions. Some these two questions you know give you the basic uh, idea कि okay fine ये दोनों criteria पूरा हो गया. But when you actually trace through the generation, like one generation after another, then only you have to you can confidently say that whether the phenotype is X-linked, Y-linked, autosomal dominant, recessive. Okay, so first you have first it is easy to just have to look these two things. Then you have to trace the phenotype and see whether whatever you have answered or whatever you have made the observation through these two questions, whether it is matching or not. Okay, now let's go through few pedigree examples. Okay, so this is a pedigree chart that is given to you. Okay, uh, take few minutes and try to understand what is the inheritance here, whether it is autosomal, uh, whether the genotype is dominant or recessive, or whether it is related to the sex chromosome. Take few minutes, like 2 minutes maybe, and then I'll start explaining this. If you have a pen paper with you, or you just have a quick look at the pedigree. Okay. I request that while I'm solving this question, I would request your participation. Okay, I'll ask you a few questions. If you know the answers to those questions, feel free to unmute yourself and answer. Um, okay. If you are done, um, let's work it out. Okay, and try to understand what is the pattern of inheritance here. So first, look at the first generation. The mother is affected, right? Then there are one, two, 1, 2, 3, 4, 5, 6. There are 6 children, and out of 6, 3 are affected. So 50% of the children are affected, and the mother is passing on the gene to both male and female. So till now, I am thinking that it is an autosomal disorder. Okay, it is since the gene is present in the autosome, so it can be passed to both his sons and daughters. Okay, and 50% of them are affected. So I'm guessing that the genotype of the affected individual um, is heterozygous, where capital A is my affected allele, which is causing the disease. Okay, and so going with this assumption, I'm simply writing the genotypes of the affected individuals. Right. Next, when we go to the second and third generation, we see that this guy married a girl, and the two of their children are now affected. And again, male, both son and daughter are affected. So I'm guessing that this is again autosomal. The disease is not there here, uh, even though um, the father is having the disease, the mother is not affected, but none of the children are affected. Okay, so here it is not getting passed. Here the daughter is affected. Here also normal, and the wife was affected. It was not in the family. So in every generation, this disease is occurring. Okay, and both male and female are affected. Like in this case also, 50% of the individuals is affected. Like here one daughter is affected, here one son is affected, here also 3 of the children were affected, rest 3 were normal. So I'm guessing that this will be autosomal dominant type of inheritance. Like this pedigree is showing autosomal dominant inheritance. So does everyone think that it is correct, or does anyone have any other conclusion? If you are in the meeting and listening, and you have tried it yourself, please unmute yourself and speak.
Yes, ma'am, it is autosomal dominant.
Okay, so according to Shweta, it is also autosomal dominant. Thank you, Shweta. So let's see, let's check. So yes, it is autosomal dominant, and this pedigree is whether, like, let's see, these are the characteristics of autosomal dominant in like inheritance. Let's see whether these points are being satisfied or not. So the affected individual usually have at least one affected parent. So you can see the affected individual, they had one parent, right? So it is evident from here. Then it can be present in either sex. Yes, it is matching. It is transmitted by either sex. So father can transmit as well as mother can transmit. Here mother was transmitting. And there is a 50% chance of being affected. Like the child has a 50% chance, right? So yeah. So if we do genotyping, um, right. So this will be AA, normal type, okay? And this is also AA, normal, normal. Mm, this is normal. This is normal. and this is A is normal. So autosomal dominant. So it is so here it is referring to heterozygous, right? So it can have AA, this AA normal hoga, and this will be AA. Okay. So you're understanding, right, how I'm writing the genotyping. So if you don't recall this, like do the Punnett's diagram and do all the combinations of all the different alleles. So it was like you were taught in class 12th. Okay. So it is using that principle only the genotypes are being assigned. Now let next look at the another pedigree. Now you're given this pedigree in front of you. And again, someone has asked you what is the pattern of inheritance. So as I said previously, you should look for whether it has skipped or not, skipped a generation or not, whether both male and female are getting affected. Okay. And then if these two questions should able to give you the answer, but sometimes you may not get the correct answer. So you have to look for something else as well. Okay. I'm giving you a few seconds, like few minutes, maybe have a quick look at this pedigree, and then I'll start, going through it, and let's match whether we are correct or not. Okay. Um, if you have, analyzed it yourself, whatever answer you have gotten, let's check whether it is correct or not. So here, none of the parents are affected. Okay. But one child is affected. So either the mutation has arrived for the first time, or the affected allele is of recessive type. Either of those possibilities are there. Then these two children are normal, second generation. Then third generation, we see that two relatives have married, right? Contiguous mating is there. So, and then this, as a result of this contiguous mating, a child affected with the disorder is born. So this is the few, feature of this pedigree is this contiguous mating, and either of the sex is affected. The skipping is there. So I feel like this is autosomal recessive type of inheritance. What about you guys? Like, can you are you able to draw the same conclusion, or your conclusion is different?
No, ma'am, it is autosomal recessive.
Okay, thank you, Shweta. So let's check. So yes, it is autosomal recessive. So what we see here is the affected parents are usually born to unaffected parents. So here, this daughter, she has unaffected parents, correct? Then parents of affected uh, people are usually asymptomatic carriers. So in order to see this, we have to write the genotypes. So normal people will either having heterozygous or homozygous. Okay. Here, no, homozygous is not possible, right? Because it is autosomal recessive. For recessive gene to express, there has to be homozygous condition, right? This will be the genotype of the parents. Now, this will be genotype of the children. This person has married to this person, so she can be either capital A, or this, right? So for affected child to be born, the parents should have a genotype of like this, correct? And what else? We can't predict the genotype of the boy who married into the family, and because of which we don't know the genotype of this. So yeah, so while after genotyping, I'm able to see that the parents of the affected individuals are usually carriers. See, the father and mother, they are carrying one copy of the recessive gene, that is the effect which is causing the disorder. Then there's an increased incidence of parental contiguity. So this is a unique feature of autosomal recessive disorder, that there is contiguous marriage, and it does affect both sexes. And the birth of an affected child, after the birth of the affected child, each subsequent child has 25% chance. So here you can see, due to the crossing of A and A, out of 4 children, only one is getting affected. So there is 25% chance. Now, this is another pedigree that you are given. And again, I ask you to go through this pedigree and like guess what will be the pattern of inheritance. Previous two examples, previous two pedigrees must be clear to you, right? We could easily determine how they were autosomal recessive and autosomal dominant. I hope the concept is clear to you all.
Do ma'am, daughters are carrier and sons are affected, so it could be X-linked recessive.
X-linked recessive. Okay. Okay. Thank you, Shweta. So only Shweta is doing, or rest people are also doing it? What about Amardeep, Ricky? Are you guys doing it?
Yes, ma'am.
Okay. So do you agree with Shweta?
Yes, ma'am, I agree.
Okay. Okay. Thank you, Bhavya. So let's see whether Shweta and Bhavya, they are correct or not. I also try it myself. So here, the mother is a carrier, okay, and in the second generation, none of the people are affected. Third generation, the son is affected, right? And in fourth generation, again, a son is affected. So what I'm saying is only the males are getting mainly affected, and females are the carriers, right? So you were saying X-linked, right? So the males will have one copy of the X chromosome, and if that X chromosome have the affected allele, then it will get expressed. So if that, even if that affected allele is recessive, it will still get expressed. But the females, they will have both copies of X chromosomes, right? Uh, but since if the gene is recessive, the another copy, that is normal wild type copy, will suppress the expression, so they will simply become the carriers. So I also feel that it will be X-linked recessive. Now let's check. Okay, let's see whether our conclusion is correct or not. So it is true, this pedigree shows X-linked recessive type of inheritance. So as I was saying, that it affects mainly males, right? But it can affect females if the phenomena of X-linked X chromosome is inactivated, or X inactivation. So in the females, there are two copies of X chromosomes, right? So and both the copies have the same genes, right? Uh, same alleles. So what happens is we don't need double expression of one particular gene to prevent that one copy of the X chromosome is silenced. So and it happens randomly. Which one will get silenced? We don't know. Okay, the nature decides this on its own. So so if the female who has received the a defective gene from her father, okay, and uh, and by chance the normal copy of the X chromosome is get silenced, and another copy of the X chromosome with the defective allele which she has received from her father is getting expressed. So it can also affect the females as well. So so that is what it is written here in this point. Females may be affected if the father is affected and the mother is a carrier, right? And there is no male to male transmission. So I'm able to see that the father is not transmitting the defective allele to his son, isn't it? Because father will only give the Y chromosome, not the X chromosome. But he has given the defective allele to her daughter, right? But because of the normal copy from the mother, the gene is not getting expressed in the daughter. Then affected males are usually born to unaffected parents. So yeah, the mother is a carrier, so uh, the uh, the defective allele is getting transmitted to male. So yeah, the conclusion is correct. The given pedigree is X-linked recessive. Okay. This is another pedigree given for you to look at it and then guess what will be the pattern of inheritance.
Autosomal recessive.
Okay, autosomal recessive. What about others?
Ma'am, X-linked dominant.
Yeah,
X-linked dominant. X-linked
X-linked
dominant.
dominant. Okay, X-linked dominant. Thank you, Shweta. So I let's see whether and like who one of you is correct. Okay. So here, father is affected. Okay, and they have 1, 2, 3, 4, 5, 6 children. Okay, and out of 6 children there are 3 daughters. So the affected father is transmitting the disease to his 3 daughters. The sons are not affected. Okay, then the mother who is affected in this generation, uh, she is not transmitting the disease to any of her son. Neither her daughters are affected. And here then again that gene is expressed here in this generation, but the mother father, they are not affected. What about here? Here the father is affected and then again transmitting the defective gene to his daughters, and then daughter is affected. So I'm seeing that the father, the transmission of the gene is happening to from father to daughter. So father is having one X, one Y, right? And daughters are having both X chromosomes. Mother is again having XX. So the defective gene, if it is present in the X chromosome from the father, and if that defective gene is dominant, then only it will get expressed in the daughters. So from this, I'm also guessing that it will be X-linked dominant type of pattern of inheritance. But what about here? So here the mother is having the dominant gene in her one of the X chromosome. So another is silenced, right? So now when the daughters receive the X chromosome from the mother, so it might have happened that only that copy of the X chromosome which is not having the defective allele is passed on to the daughter. Another copy, the normal copy, is also passed on to the daughter. So daughters now in this generation have become normal, right? Uh, but or in this case, this lady, she might be a carrier of this X chromosome carrying the defective allele. Here, this lady, because of which she is transmitting the defective allele to daughters as well as the son, right? So let's check whether my guess is also correct, like same as Shweta's. So yes, it is correct. The pattern of inheritance is X-linked dominant, and these are the features that should be present in the pedigree. It affects either sex, but more females than males. So both male and female are showing the phenotype, but it's more females. एक दो तीन चार पाँच छः. So yes, in the pedigree, more females are getting affected than males. So even so this is the X-linked dominant. Uh, the homozygous pattern of both XX, even though it is dominant, so the dominant allele is getting expressed. Now, usually at least one parent is affected. So in this case, neither of the parents were affected, right? So here maybe X inactivation thing was happening, because of which the mother, she was the carrier, but she was not showing the phenotype. The child of an affected female, regardless of his sex, has 50% chance of being affected. So here, so in this is true for this family. The mother was affected, and almost 50%, 50% of the children were affected. Now, all affected males, for an affected male, all his daughters, but none of his sons are affected. So here, the father is only transmitting the affected allele to his daughter, and because of which all their daughters are affected. Here also, the same is true. So by thoroughly going through the pedigree and recalling the conditions for the each type of pattern of inheritance, we can guess or we can understand from the pedigree what kind of pattern of inheritance it is. Now, this is quite a rare type of inheritance, which is Y-linked inheritance, and it only runs through males, because father can transmit the Y chromosome to their sons. It never goes to daughters. So if you are given this kind of pedigree where only males are affected, and affected fathers will have affected sons, so this is a Y-linked kind of inheritance. Okay, so till now, if anyone have any query in understanding the pedigree, like how to analyze a pedigree, please let me know. If not, there is a question for you guys. So this is a question for you. Please have a read. The question says that these are few of the symbols given, and you have to identify which symbol stands for what. It's a very easy question. I'll just move on. I'll not waste too much time in explaining this question. Uh, so what is the correct answer? I know you guys know it.
Option D.
Yes, yes. The correct answer is option D, right? This is unaffected male, this is deceased, this is deceased, right? This is female, this is the proband male, and this is the gender unknown. The correct option is D. Right? Now this is another question for you guys. And it is based on whatever we have discussed till now. We have to analyze this pedigree given here, okay? And understand like what is the mode of inheritance.
Ma'am, option D.
Okay, correct. You're correct, Bhavya. The correct option is D. So how did you know that it is X-linked dominant? Bhavya, can you explain?
Yes, ma'am. As the disease is shown in both the in the second generation in both the males and females, and it's like equal in the males and females, and it is transmitted in the third generation also, and same in the fourth generation, and and the parent female is affected, so one of the X is showing its characters in the next generation. So that's why the X-linked dominant.
Okay, okay. Another feature that you can see that the father is always transmitting the allele to their daughters. This is also happening. Okay, so that is a characteristic of X-linked dominant. So you're correct, Bhavya. Well done. Okay. So next, we'll move on in understanding that whatever we have studied, right? Uh, Autosomal, then in X-linked, then whether it is recessive or dominant. So these are often, challenged with few complications, right? Few complications. So first complication is the affected person, even though have the allele that is causing the disease, but that person is not showing the phenotype, and that is called penetrance. Means the disease has not manifested in itself in the individual. That is the incomplete penetrance or non-penetrance. Okay, so this is a question which is based on this theory. Okay, so you are given a pedigree and you have to first understand what is the mode of inheritance, okay, and then calculate what is the penetrance percentage. Please try it yourself, then I'll move on to explaining it.
Ma'am, can you please explain the meaning of incomplete penetrance? I still didn't get.
Okay, okay, thank you. I'll definitely explain. So what happens is, in the family, there's an individual, okay, and that individual, in the pedigree chart, when you place that individual, you can see that that person is supposed to have, like, that person have the allele that is causing the disorder, okay, but it is not showing the symptoms. That person is not showing the symptoms of that disorder, but another sibling of that person also has the allele for the disorder, but symptoms are there for him, okay. So when this kind of a thing happens, and you can see it within the pedigree, okay, one generation after one another generation, like given here, okay, so you can understand that if you write the genotypes. So when you write the genotypes, you will see that even though that person is having the affecting allele. Okay, now, right now you don't know exactly whether it is dominant or recessive. You will only get to know when you write the genotypes, but you will see that even that the person is having the affecting allele, that person is not showing the phenotype, and it often happens in autosomal dominant kind of a characteristics. Okay, so why autosomal dominant? If you look at this pedigree that given, which is given in this example, you will see. Okay, try to first understand what will be the pattern of inheritance, okay, in this pedigree, whether it is autosomal dominant or not. Okay, and if it is autosomal dominant, you have to calculate the penetrance. If it's still not clear, don't worry, I'll go through the question. It will become more clear. If it is clear to others, please try to solve the question yourself. Okay. Um, can anyone guess what will be the answer to this question? Could anyone guess?
C.
Come again. Can you repeat what you just said?
Option C.
Option C. Okay. Thank you. So let's see whether you're correct or not. So first we have to check what is the mode of inheritance. So here father was affected, okay, and then transmitted the disease to his son. What about others? Whether they were affected or not? No. All four out of four, only one child was affected. Okay, and then this father transmitted the disease to his daughter. So both male and female are getting affected, and it is not skipping any generation. So this might be a dominant allele. Okay, this might be dominant allele as it is not skipping any of the generation, and also both male and female are getting affected. So this might be autosomal, which we have seen in a previous example. So this autosomal dominant is there in A and B. Now you have to calculate the penetrance. So for doing that, you have to first write the genotypes. What is the possible genotypes of the affected and the unaffected individual? So autosomal dominant have, right? So let's say the dominant allele, which is causing the disorder, I'm representing with capital A, okay? And the affected person can have capital A, right? Or his genotype can be capital A, small a. And the unaffected person can have genotype small a, okay? So this lady is unaffected. This for this person, right? Right now I'm writing just capital A or small a, right? Because I'm not sure right now. Now none of their children are affected. Okay, so out of four children, only one is affected. So this genotype won't be there. It will be capital A, small a, because of which the unaffected people will have AA and the affected person will have capital A, small a. Okay, now look at this family. Here, normal individual will have AA, okay, and when you cross AA and A capital A and small a, we can get this affected. So the genotype of the affected individual right now is coming to be capital A, small a. Okay, here, so the same will be true here as well. Till now, we saw in two families, AA, correct? So the unaffected individual is AA, right? She has married into the family. So the assumption that 5 will be AA might be wrong, you know. This individual 5 might be having must have the affected allele, the dominant affected allele, but it is not showing the phenotype. So this person, because of this person, this is the basically the penetrance, that this 5 2 5 like she has the affected allele capital A, but she is not showing the phenotype, disease phenotype. Okay, so for calculating penetrance, first we have to see how many people are actually showing the symptoms. One, two, three, four, five. 5 people are showing symptoms. And then divide it with how many people are there in this pedigree with the genotype capital A, small a. So 1, 2, 3, 4, 5, 6. So 6 people are having this capital A, small a. Okay, then you divide it with, so multiply with 100, and the answer would be, you can get a calculator. So I have calculated previously, the answer is 83.3. So the correct answer is C. So now is the concept of penetrance and how to calculate penetrance, is it clear to all?
Yes, ma'am.
Okay, great. So let's move on. So this is one of the complications which is seen in analyzing the pedigrees. Okay, another complication is variable expression. Okay, so what happens is, a person who is carrying the defective allele. Okay, so in a family there are various people, right, who are carrying the defective allele, and the degree of expression might be different in all of them. Few people will be showing very severe symptoms, but there might be few who symptoms are quite moderate. Okay, and like this, like quite moderate, or there might be few who are asymptomatic, means disease has not at all shown itself. So what was happening in the previous example, right? For the individual, second 5, right, second generation, the 5th individual, it was happening for him, but he was a carrier, he was asymptomatic and he was not showing any expression. So this kind of degree of expression can exist for particular disease, because of which we can get wrong results while analyzing the pedigree. Then another complication can be that there might be complementation. Okay, so what is complementation? What happens, suppose this is a mother, okay, and she has married someone, and she comes from a different family. Okay, she has married into a family, and there is one gene, this capital A denotes a normal gene, but she is having a disease because of mutation of another gene, BB, small b. Okay, and this guy, okay, he belongs to, so she has come from another family, and he is from another family. Okay, and now for him, he also has a particular disease, but the mutation is not present in AA, but the mutation is present in another gene, so which I am representing with capital letters, capital A, capital B. For him, the another gene A is normal. So when these two people have children, okay, uh, the effect, like say one daughter, son, daughter, okay, all of them, they are normal. Why? When you draw Punnett square, okay, you can guess that there can be genotypes like capital A, small a, capital B, small e. Okay, so like this, you can get various kind of genotypes from the Punnett square, which shows that the mutation in one gene, here B, or mutation in A, is rescuing the mutation or the phenotype, whatever is possible, in another gene. So mutation in one gene rescues the phenotype due to the mutation in another gene. So this is possible when we are talking about polygenic disorders. Okay. Uh, so this is what is called complementation. Is it clear? Okay. If any point till now is not clear, you can like unmute yourself and ask. Okay. There's no problem in doing that. So another complication can be the affected gene might be imprinted. Okay, so we in the previous few minutes I was explaining, right, what was imprinting. So the certain genes are linked with one of the parents, right? The the gene will only be expressed if it is inheriting from the father. Okay, same gene copy from in the mother chromosome will not be expressed. The vice versa is also possible. So this imprinting can also, play a role. Um, you can give you bit complication, makes bit things complicated when you are doing pedigree analysis. Then there is something called anticipation. So what happens that the phenotype which is resulting from a particular defective allele, uh, when that phenotype will start, okay, the age of onset, it starts decreasing, and as well as the severity means the symptoms become more deadly, more lethal to that person. That severity also increases. Okay, so if one particular disease started its onset in the first generation, like say in grandfather, it started at the age of 77. Okay, now his son or his daughter started having the symptoms for the same disease, say at the age of 40. Okay, and his daughter or son started having the symptoms of the disease at the age of 20. So like this, the age of onset is decreasing, and also this disease is becoming more severe, and like making the lifestyle of the person more different and difficult, and increases the risk of death. So this kind of trans inheritance of a particular disease also complicates when you are doing pedigree analysis. So these are few other complications. Is till now, if there is any question, do ask, as I'll move on to another topic. Is there any question?
No,
Okay, thank you. Um, so the next, I'll now briefly explain these terms, right? So which if any of you are from biotech background, you would be very much familiar what these terms mean. And these are, you can say, the tools, or you can say that they are the methods by which we can identify the defective gene wherever it is present in the genome. Okay, and make conclusions, okay, whether or not this gene is actually causing the disease, etc. And this is a very tedious process, and it takes years to finally say or conclude something with confidence. So if you guys are aware, okay, then do answer. Okay, so what do you mean by restriction enzymes?
Some
Cleave the
enzymes
DNA
that
at
recognize.
specific sites.
Sorry, there was some overlapping of voices. It was not clear to me. Can you repeat the answer?
Ma'am, enzyme that recognizes and cut DNA at specific site or sequence.
Okay, you are correct. So whatever Shweta has said, I'm just repeating. So restriction enzymes are basically endonucleases. Okay, endonucleases means they will cut the DNA or RNA backbone. Okay, and these endonucleases will cut within the DNA sequence, and these restriction enzymes, they are derived from bacteria like E. coli, etc. And people have found out that these enzymes have the property of cutting the DNA sequences at unique sites. These also called restriction sites. So people have used this unique ability in forming recombinant DNAs, right? And when you have cut to you have made a double strand cut of a DNA, so and you want to ligate, okay, you want to join again, say you want to join them again, so then for that you need to use another enzyme that is called DNA ligase. So DNA ligase is like a glue, okay, that joins together two fragments of the DNA. Then what are plasmid vectors? Shweta, would you like to try? Or anyone else? What do you mean by plasmid vectors? Where are they present? How do they
Yeah,
look like?
circular DNA used to transfer part of genetic material, circular DNA.
Okay, so you are correct. So these are circular DNAs, which are usually found in prokaryotes, right? Like bacteria and all. And people have identified that there are certain, regions like ORI, then there are restriction sites and there. So we have harnessed those properties for our convenience, and we use plasmid vectors for cloning a certain gene of interest. So they are very useful for the process of DNA cloning. So what is DNA cloning? What happens is that you take a gene of interest from the from particular organism, and you want to make several copies of it and keep it with yourself in a stable form. So people then ligate that gene of interest into the plasmid vector. So they look something like this. If I were to draw, they look something like circular DNA, right? They look something like this. And say here you have ligated your gene of interest by cutting this circular DNA using a restriction enzyme. And there are ORI, or the origin of replication. Okay, and this ORI, from this point onwards, the replication of the entire DNA sequence will begin. So this kind of a structure is then transfected into bacteria. Generally, E. coli is used, okay, bacteria, and within the bacteria there is enzymes available, right? Uh, Recombination enzymes and all. So these enzymes will start the replication process, and as the bacteria is dividing and making daughter cells of its own, your gene of interest is also getting distributed in those daughter cells. So in this way you are making clones, or we are making copies of those of your gene of interest which are present in the different bacteria colonies. So in this way you can safely store and use that for any other purpose later on. So now, what do you mean by genomic library? What does genomic library mean? This is also a very easy question. I'm sure you, like one of you, must know what is genomic library. So please don't hesitate to share with everyone. Shweta, would you like to try?
Basically, it represents almost all segments of chromosomal DNA.
Okay, so you're correct. So what happens, like for any living organism, you can take a human cell or bacteria cell or plant cell, anyone. You take the cell, okay. Then from the nucleus contains your genomic material, right? You have to take those out. So there are protocols available for isolation of this entire DNA content, and then you use these restriction enzymes, okay, and make fragments. Now these fragments can be you called as gene of interest, and ligated within plasmid vectors, writing as PV. And these plasmid vectors then transfected to bacteria, and then you have done what is called DNA cloning. In this way, you have made the DNA fragments available in form of separate bacterial colonies. So now you can do what do you say, you can store them separately, you can make different fractions, like this fraction of the bacteria has, you can label one fragment as one, this fraction of bacteria have two. So in this way you can create bacterial colonies and clone those fragments and create a library for yourself. So if you want to study what all genes are present in say fragment 1, you will take the bacterial colonies and then again lyse the cells, take out the DNA fragments, and then do the further studies. So, every time you don't have to go back to the plant cell or human cell from which the genomic material was taken. You can just refer to this library that you have created for yourself, and it takes months. Okay, it takes months to make a genomic library. Then another thing is called cDNA library. So cDNA, what does cDNA stands for? cDNA stands for complementary DNA. Okay, so let me show you a diagram to explain this. So in this diagram, which I've taken from a science article, so here they have shown how a cDNA is made from mRNA. So first you have to make the fragments. For cDNA, you need the mRNAs, that is the expressed genetic material, which will ultimately give you the proteins. So cDNA are made from the mRNAs. So first you have to isolate all the RNA content, total RNA content from the cell, and load it into an elution column. Now, you know the signature, a unique property of mRNA is that they have a poly-A tail. Okay, so you have to use that for separating mRNA from other types of RNAs. So the elution column that you see here, it has cellulose beads. Okay, and to those cellulose beads, they link oligo-DT. It is a short sequence of only T, which will then show complementary binding with poly-A tail of your mRNA. So here exactly what is happening is that only the mRNA which is having the poly-A tail. This is the collection of all the RNA in the cell, and the poly-A tail is present only in mRNA. Now this is loaded into this elution column which you made previously, and the mRNAs with poly-A tail, they have complementarily a bond with the oligo-DT. Okay, and when you then pass the entire solution containing RNA through this elution column, you will see that RNAs, different RNAs like siRNA or tRNA, rRNA, which didn't have poly-A tail, they will be passed through, whereas the mRNA will bind to the cellulose beads into the column. They will be retained, okay, through hydrogen bonding. You know that, right? Then you use a buffer in order to break this hydrogen bond and then elute out only the pure mRNA which you have gotten. This is how you have purified mRNA from the total RNA content of the cell. Now this is your mRNA which you have eluted. Next, you will do the process for making cDNA or complementary DNA. So why people choose to use cDNA, or why people choose to make a cDNA library, not mRNA library, right? It is because mRNA, the RNA structures, since they are single stranded, they are very unstable. They denature, okay, very quickly. So for long term storage, um, people make DNA. DNA is more stable than RNA. You must have studied it somewhere in your class 12 or even your graduation period, right? So that's why cDNA is more preferred than storing simply the mRNA. Then what you have to do, you have to again use the oligo DT, which you have used in the previous step, and this time it will be used as the primers. Okay, so as you can see here, this primer is attached at the 5' end and giving OH group in the 3' end. Okay, so you need this OH group to which subsequent nucleotide molecules will be added, right? The synthesis will happen from 5' to 3' end. So the enzyme reverse transcriptase will be used now to make a DNA sequence of where mRNA will be used as a template for synthesis of this DNA sequence by the enzyme reverse transcriptase. Now, after reverse transcription, this is your cDNA which is ready. This is your mRNA. So this is a hybrid structure that is made here, right here. Then what you have to do, you have to use a RNase enzyme which will partially cleave the mRNA, okay, at certain places. And but some part of the mRNA, like this, some part of the mRNA will be intact, because we need those mRNA to act as primers. As you know, synthesis will only happen the new because in place of mRNA you will be synthesizing a new DNA, and it will only happen from 5' to 3' end. So if you remember the lagging strand of that is there in the replication fork, so there fragments are formed, right? Okazaki fragments, if you remember that concept. The similar thing is happening here as well. So the small undigested mRNA fragments will be used as a primer by this polymerase enzyme DNA polymerase 3 in order to add nucleotides and then create a new DNA strand. Okay, and it will proceed like this. Okay, from 5' to 3'. These are the DNA polymerase enzyme. And later on, these small fragments, they will be ligated by the DNA ligase. That is the function of the DNA ligase, to join two DNA fragments. So in the end, you will get a double-stranded cDNA fragment. Okay, for your particular mRNA. Now this cDNA can be then used for DNA cloning. You can use it as your gene of interest, insert it into a plasmid vector, okay, transfect a bacteria, and store it safely with you for further application. Is the process how cDNA is made is clear? So you can do for all the mRNA that is present in the cell and create a cDNA library. All right. Does anyone have any query in this point? Okay, great. Now, your cDNA or your normal DNA, genomic DNA is now ready with you, and now you want to study, say, what all genes are present. Till now, you don't know, okay, your fragment, the sequence of your fragment. Uh, it can have the repetitive non-coding sequence as well, or it can have gene or non-coding sequence. Now you need to know what is actually present in your fragment. So you can first amplify that sequence by using the process of PCR, polymerase chain reaction. Okay, so here you must be aware, you have must studied in your BTech or in class 12. So what happens? Let me draw and explain. I think that will be more clear. So this is your, say, your DNA fragment, okay, and this is a double-stranded DNA fragment. First, you have to denature it, and this process is called denaturation. Okay, and it happens at a very high temperature. Okay, like at around 90, 96 like temperature. Okay, and what happens after denaturation? These two DNA strands, they separate from each other. The hydrogen bond, which was holding these two DNA strands together, they have now been broken. After denaturation, you will be adding some RNA primers, or they can be forward and reverse primers. One primer can, so here is a denatured DNA. One primer can bind here, one primer can bind here. Here I'm not mentioning the direction, but you can like say, let's mention it. Why not? So this is say 5 prime to 3 prime. This is 3 prime to 5 prime. So here primer will be from 5 prime to 3 prime. Here 5 prime to 3 prime. Okay, so the primers which are binding complementary to your sequence. So, and then this is your annealing step. Step is called annealing. Now, when you have annealed your primers, you have to amplify. That is, you want new DNA fragment, that is, it should be synthesized by the process of DNA replication. You must be aware. For that, you need a polymerase enzyme, right? So because of that, after polymerase enzyme has done its job, you get two separate DNA fragments. So from one DNA fragment, you're getting two. This is how the PCR works. And so initially, so this helps you to get a large DNA sample. DNA sample is now large, which you can again store and do other analysis with it. Okay. So once you have done PCR, you have amplified your DNA fragment. Now you want to see two things. Okay, what is your size? If you want to know what is the size of your DNA fragment, then you have to use this method, the agarose gel electrophoresis. Okay, so uh, if you recall, an agarose gel, DNA is loaded in some wells. Okay, you have to make that wells in the agarose gel, and by applying electric current, right? So DNA is negatively charged, right? So from negative to positive charge to cathode to anode, the DNA fragments, they migrate. And because this agarose gel is polymer, it's a polymer, it has pores in it. Okay, so if the DNA size of the DNA fragment is quite small, it will it is easy for it, it is more easy for it to pass through the pores. So the small DNA fragments you will get towards the end, whereas the big DNA fragments, it is difficult for it to pass through the pores. It will be at like somewhere up. It may not able to migrate so much towards the positive terminal. So this is how you do agarose electrophoresis, electros gel phoresis, and based on the size of the DNA fragments, the fragments gets distributed, like you get some this kind of a pattern. Okay, so from this you can and if you run a ladder or a scale, you can say it is a scale actually, you can then use that ladder to determine the what is the molecular weight or the size of a DNA fragment. So if the ladder is showing 0 to say 40 kb and your DNA fragment is towards the end, so you can now say that this my DNA fragment is of 40 kb. Okay, now you still don't know what is the sequence. So for sequencing, you use the method Sanger sequencing. Are you guys aware what is Sanger sequencing? So for so long I'm talking. I want now one of you to speak up and tell a few things about Sanger sequencing, if you're aware of it. Anamika or Ricky, Amardeep, any one of you, if you guys aware of it. Have you heard the term Sanger sequencing? Shweta, तुम्हें बता दो. Okay, who is speaking? Please go ahead.
Sanger sequencing is dideoxy sequencing in which dideoxy nucleotide will terminate the sequencing, and at that point we got to know which nucleotide is terminated, and thus we get the fragments after electrophoresis, and after based on electrophoresis we get to know the sequence.
Okay. So Amardeep, you are correct. So what happens is when you take the DNA fragment, correct? I'm just repeating whatever Amardeep is saying. So you take your DNA fragment, and while you're doing PCR, instead of adding normal oligo-DTs, okay, you add labeled oligo-DTs. So people add radio labeled, okay. Radio labeled means in presence of gamma rays, x-rays, जो भी, then you will get the image. Okay, so you add radio labeled nucleotides in your PCR mixture. So when the PCR is happening, when you're getting the amplification, okay, you, and also these radio labeled DNA fragments are such that they don't allow further addition of nucleotides. So suppose this is my DNA fragment, okay, and at this stage, your radio labeled like radio labeled nucleotide is added here, okay, but here this nucleotide, it doesn't have the OH group, because of which the DNA synthesis after addition of this nucleotide is terminated. So special name is actually there. I think DD double deoxy means it doesn't have OH group at the 3' end, NTP. It is called DDNTP. So because of this characteristic, no further addition of nucleotides happen after this point. Okay, so you can have for A, T, G, C. Okay, now wherever this DDA, DDATP, DDTTP, GTP, DDCTP, wherever they're added, the further addition of the nucleotides has stopped. Now you take that PCR mixture at the end of the PCR, you run in gel electrophoresis, and you then see how the fragments they have migrated in the agarose gel, and you will see that based on the size you will get like this pattern. Okay, from seeing this, you can then you then have to read it. Okay, you have to read. So say you have a mixture, a PCR mixture where only A was added initially. Okay, so for that PCR mixture you run in one lane. Then you take another PCR mixture where only DDTP was added. So you run that. Similarly, ATGC. So for each PCR mixture, you get one column in your agarose gel. And then you have to read from the bottom to top, because at the bottom, the smallest DNA fragments are there. Okay. At the top, the large DNA fragments are there. So it means if you're reading from the bottom to top, you're reading from 5 prime, okay, to 3 prime. So so in this way, when you read, you will get your sequence. Like say it starts with A or T, with G, C, like this, you can do it. So there was one example. I think there was one question in the week 2 assignment based on this, where you had to interpret what is the DNA sequence. So this was based on this concept only, that you had to read your DNA sequence. You have to read the agarose gel, okay, and find what is the sequence of your DNA. Am I correct? Was it in your assignment, or I have read it somewhere else? Can any one of you can confirm?
It is there.
It is there, no? So I think if you have like understood that this concept, or you have read it somewhere else previously, you could do that question. Okay. If anyone has any difficulty in understanding that question, maybe we can discuss it here. Okay.
The sequencing once more in a short, in short.
Okay. So in Sanger sequencing, what is happening is you take your DNA fragment, okay, and you make different four PCR mixtures, but instead of adding your normal nucleotides, you add DDNTPs, where at the 3' end the OH group is missing. Okay, so you have four types of DDNTPs, A, T, G, C, and in one mixture you add this, second mixture this. So you have four types of PCR mixture, and then you do the simple normal PCR. So wherever say ATP is added in the amplification, after that whatever sequence was there in the template was not amplified. Okay, so in this way, for every for wherever uh, this DDNTP was added, after that the sequencing didn't happen. Then after the PCR, you run it in gel electrophoresis, okay, and you observe the columns. You make one in one column you run the PCR product that you where DDATP was added, and the PCR product where DDTTP was added. Like this you get four columns, okay, and you observe like how the DNA has migrated based on their molecular size, right? And then you read from the top from the bottom to top, where because at the bottom the small DNA fragments are there, and when you read it, you are reading in the 5' to 3' direction. Okay, so the smallest fragment is your A. Okay, say I'm trying to draw it clearly as much as possible. So this is A, T, G, C. So this is the smallest A, okay, and then उससे थोड़ा बड़ा T के ऊपर थोड़ा G is there. This is another lane, and this is C. So you read like your 5' end first will be your A, okay? And then उससे ऊपर the second band is for the T, then third band is for G, then fourth is for C. Like this, you read your means you read from the bottom to top, okay, based on where the DNA band is present. So in this way, you can then deduce what is your DNA sequence in 5' to 3' direction. Is it clear?
So this PCR, this gel readout comes after it is completed the agarose gel electrophoresis, right? After finishing of the PCR process.
Yes. Your PCR product is run in gel electrophoresis.
It's loaded into the well, and it is after running.
Correct. Yes, you have to supply the voltage, and it will run. Then you have to read it. Okay.
From this
Yeah.
gel pattern, Dr. Sanger's sequencing, if gel is sequenced and it is being shown, so how do we come to know the cDNA from the gel?
The cDNA.
From the gel pattern.
So cDNA is what? A normal DNA, right? It's just normal DNA fragment. So see, cDNA, the sequence, you have to check in cDNA genomic library, okay, or you can get the sequence first and then make a complementary, okay. It will be complementary to the sequence of an mRNA. So if you know what all mRNAs are present, and if you know the sequence of those mRNAs, okay, then for the DNA, cDNA, you don't know, okay, you don't initially that whether it is cDNA or not. So you have just the DNA sequence. Whatever the complementary of this sequence. So if it is A, then T, A, G, C, you write the complementary, but for mRNA, where T is replaced with U, and then you match in mRNA sequence which is available with you in the literature, or you have made a previous library. Okay, and then you can say ki, yes, this is complementary to the mRNA sequence. Okay, so this is your now cDNA. The sequence of the DNA fragment which you are obtained, it should be complementary to the mRNA sequence, which is, this is what I explained. What happens, like how you produce cDNA. This is the fundamental of how cDNA is produced. It is produced from the mRNA sequence. You have to just use that.
But suppose the result is being shown and the just gel pattern, if we read out, so can we get the cDNA out of it? Suppose like the gel pattern which you have just drawn these two. So can you find out, can we find out the gel pattern, the cDNA from the gel pattern? If it is not
No,
mentioned.
no, you cannot say that it is just any other DNA sequence to you. You cannot say ki it is a specific kind of a DNA like cDNA. You have to
Suppose to
yeah.
so suppose we have to get the mRNA this from the sequence which is being shown, the mRNA sequence, and if they have shown, if they have asked for the cDNA, from the cDNA you have to tell the mRNA. Can we do that from this?
You have your mRNA sequence with you, right?
No, I don't have, but they have mentioned just they have given the sequence, and they have
Okay.
asked to give, they have asked to find out the mRNA from the cDNA, but we by ourselves we don't know. Just we have got the sequence. We have to find out the cDNA as well as the can we do that?
Yes, we can do that. I think it is in your example. If you want, I can solve it for you. And
Do not.
solution is
I want you to solve this, but it just we need to learn the process how to do it.
okay. So let me tell you the process. You can do it. It is not that difficult. Okay. So your Sanger sequence. If you are not able to understand my handwriting, please let me know. Okay. So
No,
you're
it's fine.
okay. So you're given a gel, right? And this is the gel you're given, and these are your columns for ATGC, and the DNA bands are there at different positions, and you're reading the DNA bands from here bottom to the top in this direction. And say this is, and in you have read out like this A, then G. See, so if A is at the एकदम bottom में है, so थोड़ा सा ऊपर है G, then you will read G, not T, because T is at the top, right? Then C is, C यहाँ पे थोड़ा G के थोड़ा ऊपर है C, then C से थोड़ा ऊपर है T, okay? Then इसके बाद उसके ऊपर है G, and this is C. So this is the sequence, any sequence in a DNA sequence that you have obtained, and it is in 5' to 3' direction. Okay. Now now in the question they have said, they have mentioned, right, according to you, that it is cDNA, correct? They have mentioned that ये cDNA का ही जेल इलेक्ट्रोफोरेसिस है, जिससे and ये Sanger sequencing करके उन्होंने बताया है, right? This is your cDNA sequence. Now you have to complementary, write the complementary. Okay.
This hasn't been said. They said you have to find out the cDNA, then you can predict the mRNA. Both needs to be find out.
ओ तो ये तुम्हारा जो सीक्वेंस जो दिया हुआ है वही तुम्हारा, तुमको जेल दिया हुआ है, राइट? व्हाट इज गिवन टू यू? जेल दिया हुआ है, राइट? तुमको ये सैंगर सीक्वेंसिंग करके इन्होंने ये जेल का इमेज दे दिया होगा इन द क्वेश्चन. हाँ, दे दिया होगा. एंड in the question they from the gel you can get this DNA sequence by reading it out from bottom to top.
So
can you do that?
we called the main problem is to understand DNA. Can we we can find out the cDNA? It's not a problem of solving the question, but how to get the cDNA? If they have said that you need to find out the cDNA from the sequential from the picture itself.
Okay, so let me find a Sanger sequence का gel for you. Okay, so I'm stopping the क्या बोलते हैं presentation. मैं थोड़ा स्क्रीन शेयर करना बंद कर रही हूँ एंड मैं इंटरनेट से एक सैंगर सीक्वेंस का इमेज निकाल देती हूँ एंड उसको इस एग्जांपल से तुम समझ जाओगे कि कैसे रीड आउट करते हैं. ठीक है. ठीक है. So I'm sharing my screen again. So this is a gel. Okay. ऐसा टाइप ऑफ एक जेल है, सिंपल है इमेज एंड ये जो जेल इलेक्ट्रोफोरेसिस किया है ये सैंगर सीक्वेंसिंग का जो प्रोटोकॉल है मैंने बताया था, यूजिंग दैट दे हैव ऑब्टेन्ड. ओके, नाउ you have to from this image of the gel you have to determine the sequence, what will be the DNA sequence. तुम्हारा यही डाउट है, राइट?
Like the
Correct?
question is framed in manner, I can tell you. So from the sequence of the Sanger sequence gel, the gel has been sequenced, and it has been found, and you are being asked to solve to get the mRNA from the cDNA, from the cDNA. But the cDNA hasn't been mentioned whether it is cDNA or the DNA which is being sequenced.
I think I think you are understanding the question wrong. The sequence that is given to you is of cDNA only. Let me is it the question in week 3 or week 4? Sorry, कौन सा
It
week
is
का
in
assignment है? कौन सा week का assignment है वो? week 2, week 3, week 3, week 3. मैं थोड़ा सा question खोलती हूँ, ठीक है?
Don't solve it.
Okay, I'll not solve it for you. ये वाला question ना, ये वाला question, second question. Okay. तो इसमें I think ये तुम्हारा जो sequence यहाँ पे लिखा हुआ है, assume that the DNA strand being sequenced is cDNA only. ये cDNA का ही sequence है जो तुमको जो दिया हुआ था.
So cDNA, so we do not need to find out the cDNA in this, right?
You cannot do that. Theoretically, it is not. Actually, you cannot do that. तुमको ऐसे 5 प्राइम to 3 प्राइम कुछ भी दे दिया जाएगा, ATGC कुछ भी, ठीक है? तुमको नहीं पता चलेगा the type. It is just a DNA sequence to you. Just DNA sequence. You can just see A है, T है, G है, C है, और वो तुम्हारे लिए एक DNA sequence है. वो क्या टाइप का DNA sequence है, वो क्या फंक्शन है उसका, ठीक है, कहाँ से आया, you don't know until you do further analysis. Okay, so वो क्वेश्चन में यार सैंगर सीक्वेंस जो दिया हुआ था वो तुम्हारा cDNA का ही दिया हुआ था, ये जो इमेज, ठीक है? एंड उसके बाद तुमको वो इमेज से यू हैव टू फाइंड आउट व्हाट इज द सीक्वेंस एंड देन डू further step. If you know the further step, then you can do it. It was not a difficult question. ठीक है, समझ में आ गया? और कुछ डाउट है?
So the mutations are being classified into many types, right? So if one stop codon gets incorporated, it totally stops the protein synthesis mechanism, and it forms truncated protein, like point nonsense mutation, and the frame shift mutation by like insertion deletion does do the same thing like this nonsense. These are called pi mutations. Right? In this sense, we do not know what kind of amino acid is gonna be made, but still it produces the protein. So it will not truncate the protein synthesis, but it will produce protein, but we don't know the function of it, right? So that's why I call it missense. So there are some other classifications of mutations which are being discussed by Professor Ganesh, uh, that the loss of function, sorry, dynamic mutations. So what are called dynamic mutations and how are those are classified? What are its types and how how those are being grouped in which manner? Like proteins, like we know, like proteins does have four types of structures: primary, secondary, tertiary. Sorry, tertiary, quaternary, and protein could be again classified according to its function, like complete proteins, incomplete proteins, those kind of simple proteins, complex protein, compound proteins. So according to solubility, right? So these are the all type of classifications we can do. Uh, it can be done actually. We have seen it, but how mutations are classified? So these some other kind of classifications like point mutations, missense, nonsense, frame shifts, and again these dynamic mutations comes into play. So what are these kind of classifications and how and why these are being classified into such kind of manner?
So what I could gather from your question that you want to know कि dynamic mutations क्या होते हैं, right? And where this dynamic mutation falls in this classification of mutation. You want to know that, right? Am I understanding your question correctly?
So I don't know exactly whether I made you understand the way I wanted, or is there any wrong in framing the question?
Arey, I think you want to know what is dynamic mutation, correct? You're
Yeah,
not
yeah, I
able to
wanted.
understand. Yeah. So in brief, okay. So I was planning to discuss this in week 3, like next week. But in brief, dynamic mutations, what happens? Um, do you understand the term non-allelic recombination?
Yeah.
Yeah. Okay. So during what is non-allelic recombination?
So
Uh,
it is just the opposite of allelic recombination, which happens between the parents. And in case of a non-allelic recombination, it's just it can be a pre-exposed condition for other diseases. Like it can be causative reagent, causative agent and event actually. Sorry, I said it wrong. It's an event which leads to a lot of diseases.
Okay, so let me explain in a better manner. Okay, so I found a definition actually for you guys for what is non-allelic recombination. So I have taken it from the Wikipedia. So non-allelic recombination, it happens during the meiosis one, where crossing over and all happens between the homologous chromosomes. But the regions where the recombination is happening, they are not alleles, okay, but they are of DNA sequence with high sequence similarity. Means wherever the crossing over is happening, you understand, right? Crossing over, uh, one sister chromatid is crossed with another sister chromatid of the two homologous chromosomes, okay, sorry, two non-homologous chromosomes. So the recombination is happening at the place where the genes, they are not alleles to each other, but the DNA sequence similarity is quite high. So because of this non-allelic recombination, what happens often, the DNA sequence get deleted from the one of the homologous chromosomes. Okay, it gets deleted, or it often gets added. Like duplication is also happening. So you can see here, it duplicates. Okay, or the copy number gets changed. It can increase or it can decrease the copy number. So because of this, uh, the region after the recombination, the final regions might have a loss of DNA sequence, or DNA sequence amount has increased, or same block of DNA sequence has repeated. Okay, so copy number is increasing for a particular segment of a DNA sequence. So in dynamic mutation, what happens is that when a particular allele, you have identified an allele which is causing a particular disorder, when that allele is getting uh, transmitted from one generation to the another generation, the allele copy means the DNA sequence that is coding for that allele, that number of copies, like say in first generation it was one number copy, second generation because of this non-allelic homologous recombination, the copy number increased. Okay, in third generation then again it increased. So you are not able to understand uh, like how many copy number is actually needed for the disease phenotype to happen. So people have seen that if the copy number of that particular defective allele it increases with one generation to the second generation, you get that variation kind of a thing which I explained in complications, what complications can happen. So one phenotype which is associated to a particular disease, it can be severe if the copy number of that particular defected allele is more, okay, in
That's
comparison to the correct, correct. No, not anticipation, variation. Anticipation, age of onset decreases and the severity of the disease decreases, but in variation the percentage of symptoms can be different, like person can have 50% of the symptom, one person can have 30% of the symptom. Okay, one can have showing all 100% of the symptom, one person is not showing any symptoms at all. Okay, so this kind of observation is actually coming from like dynamic mutation, like a few research papers are have explained this. Okay, I'll go in more detail in next week, like when we'll discuss week 3 content. Okay, but for now, what is dynamic mutation, how dynamic mutation is happening, okay, what is the consequence of dynamic mutation, in brief I have explained to you. If it's not clear, I'll repeat again. Yeah.
Loss of function, gain of function comes under the dynamic mutation part or something else?
So if we term a mutation as loss of function in terms of protein, okay, means if suppose one base change is there, okay, one base change is there, like in what happens in point mutation, because of which the encoded protein from the mRNA is not functional at all, then the loss of function is termed. The function, uh, whatever bodies have, whatever a cell functions because of the protein. So protein are the actually things that causes any function to happen. So loss of function, you are telling in terms of the protein, whatever protein is encoded from the resulting mRNA. Okay, so there are gain of function. One point mutation has happened, or one dynamic mutation, any kind of mutation which has happened in the DNA sequence, because of which the encoded protein has gained a new function. Okay, so then that kind of mutation which is resulting in a new function of a protein will be called gain of function. Okay, so dynamic mutation is not necessarily a loss of function or a gain of function. It can depend, you know, where it is happening, where the DNA sequence number of copies is getting added, whether it is getting added in the, you know, the frame, the open reading frame, in between the open reading frame, because of which the resulting protein is non-functional, so then it will be loss of function. But if the resultant protein is getting a new function, then it will be gain of function. So it can be either. Okay.
So I'm just trying to find out that how it is being, how it can be linked. The dynamic mutation, it seems to be a newer part.
New part. It's a new part, a separately new part actually.
So they can be classified into many types, or dynamic mutation doesn't have any other classifier, any other types or any other subforms. It does have only itself. It's the same. It's the only term we can use, and it is of one type only.
Means dynamic mutation doesn't have a sub classification, at least not I have read till now. But if I find any sub classification, I'll explain in week 3. But till now I can say that there is it is an individual class of a mutation. Okay, there is no subclass of dynamic mutations till now. But if I find any other classification, like subclassification for dynamic mutation, I'll explain in the next week. Okay.
Yeah. And one more thing, like for laymen or like people who are not related, or I don't know exactly, for people who ever for convenience, they can refer to Wikipedia or any other resources, whatever available, available, like on that time. But for 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 research people, or for the people who work in the field of research, so in case if they do not know anything, any term, or if they need to refer a single term, so where they might go for, do they refer Wikipedia, or do they straight away go to the library and search for the terms, how they can find out new terms?
So they can do specialized courses like this, human molecular genetics is a specialized course. Or you can they read research articles, okay, where first people discovered,
Articles,
like,
whatever I understand, research is in its own way, it's very unique, right? One research is very unique to other. It can be the same. It can't be the same. So
like whatever we are now knowing, okay, whatever information that we have known, it is not because one person only done the research. In the
yeah,
world there are hundreds of people who are doing research in the same topic. So in that same topic you can find thousands of literature, okay, and reading those research articles or review articles, okay, you can get to know more in detail. But yes,
but
from time to
immediate
time,
for
but
immediate for immediate
reference.
knowledge, for just immediate and instant knowledge, can we so do you people go to Wikipedia, or to go, or you just go
Yeah,
straight away to the articles?
nahi, when something is completely new to us, we all use Google. Just Google it, Wikipedia, या फिर any trusted website. Like you can, there are few trusted websites like Science Magazine, or uh, if there Scopus is there. Okay, Scopus is also a very good database, and as well as it has its own articles. So there we can get the definitions, like ready-made definitions, just to get a understanding of what it is. So like this, I have shown you a Wikipedia abstract, right? So at that point of time, I didn't recall, so I can just see Wikipedia. It's not a big deal. Huh. But in order to know more, Wikipedia is not sufficient. It gives a very brief definition. You have to read research articles, and researchers have to read research articles to get in-depth knowledge about any topic they are researching about. Okay.
So means I don't know exactly. For me, it takes a lot of time to read research articles too. So is there any thumb rule of getting through a research articles very quickly? Like suppose for you, if I give you one research article, right, of 10 pages for the research you are doing, or you might not be doing it, which is very new to you. So how much time it should take, or you take to cover up, or to go through the gist of the research article and to understand the matters, whatever needs to be understood? How much
Okay.
time you will?
So this is a very general question. Whoever wants to listen, they can listen. Otherwise, the time is up. They can leave the meeting. So to answer you, for me, if I want to know the gist of the research article, I first read the abstract. Okay. From the abstract, I get to know whether this article is worth reading more or not. Okay. Then and what problem they are trying to solve, I understand from the abstract. Then I often in order to understand more what they are doing, or why they are doing, or what how they are doing, I usually read the last paragraph of the introduction, where they often write, like, in this work, when you see a statement where starting with the terms, in this work we have done this, and it is often present at the last paragraph of the introduction, there they very briefly explain the experiments as well, and why they have done it. Okay. And even after reading that, if things are not clear to me, I then go to the last section, the discussion of the conclusion section. Okay. So that section also often, like, where they mention, like, what results did they obtain, whether those results are good or not, what experiments they did, why those experiments. Okay. So the conclusion, is the main thing. Now, from these three: abstract, the last paragraph of the introduction, and the discussion, I pretty much get to know what it is in the article, and then I decide whether to read the entire article or not, or you just discard it. So I then it takes, if I want to read the entire paper, it takes time. It takes time, like 4 hours, 5 hours, even whole day. The article is too long or new for me. So initial screening, I do like this.
Yeah, because for referencing and all, we need to read and again need to do you make notes while you're reading up or you're making a presentation or you're writing in some research articles. So you do that. So you read, then you make some notes, then you include that reference in your list, and then like paraphrase it. Then again,
Yeah, so everyone has a different way of doing it, and as you read more and more research articles, you will become much better. It's a practice thing that you have to do, you know. Initially it will feel difficult, but later on it will be easy for you.
yeah.
Okay. Okay, thank you for joining then. This, I think time is up, and I will be ending this meeting. Thank you all for joining. I'll see you guys in the next week with the discussion on week 3 lecture, week 3 content.
Thank you, ma'am.